Oh, perfect. Okay, so during my thesis at IBM Research and ETH, I was working with this quite unhumble task of modeling protein language and trying to apply it for different protein classification and annotation tasks. I guess most of you do not have as much biological background as I do. And you might be asking a question like, why do proteins matter? What is this, wet, weird white stuff have to do with natural language understanding or sequences of text? Well, in fact, proteins are sequences. As you can see here, oh, sorry, I could maybe turn on the pointer. Proteins are made up of amino acids, and there are around 20 of them, whereas we have 4 DNA nucleotides that get transcribed and then later translated into proteins, which facilitate most of the hard work that our cells do in the body and perform all sorts of functions. So proteins are live, and the individual amino acids come into sequence, which we call primary sequence. And then within the sequence of amino acids, and sometimes that sequence is 100 amino acids long or letters, or sometimes it's 200 letters long, there are interactions within the sequence, and that sequence can loop onto itself and form an alpha helix, which has like very low entropy and is very stable, or it can form what is called beta sheets, or B, just a flexible loop. And these structures are examples of secondary structure. And later on, the protein can fold onto itself even more and form a globular domain, become a 3-dimensional structure that performs a particular function. So this primary sequence encodes function, we say that. Now the problem, proteins are so good and they're useful, they're drug targets, whatnot, the big pharma companies, and we have so many sequences of proteins. Every day there are thousands coming off protein sequencers. However, only less than a small fraction of them actually have functional and structural annotations. And the desirable annotations are, as I mentioned, structure. How does the protein fold in 3 dimensions? If you know that, you can maybe design a small molecule or another protein that will bind to the protein and change its function. In industry, people often worry about the stability of protein as well. Will it precipitate? Will it be stable after expressing it? And often in pharmaceutical applications, you also worry whether the 2 proteins will bind to each other. So for instance, if you're developing a new cancer drug that can discover some cancer antigens that are expressed on the surface of your cells, you want to make sure that this piece of protein, this antigen, can be recognized by your immune cells that you're training to detect cancer in this personalized medicine setting. So the problem is clear, we have many sequences, but very few annotations. So we would like to predict for any new given sequence, functional and structural annotation. I'm saying that, okay, I can admit some people. I have this hypothesis and assumption that evolution is a very parsimonious and stingy driver in the nature. So evolution does not want to waste whatever it has discovered, whatever works. So for instance, if a particular protein fold performs its function well, like digesting food, some enzyme does it well, that structure will be replicated throughout life, and most of organisms will share the machinery how to digest food, or I don't know, defend against particular type of bacteria or virus. So these useful features are preserved throughout evolution, and that puts the protein sequence into this, or hidden protein sequence features into this learnable manifold that is quite small, and as I said, is probably learnable, and all of that functionality must be encoded in the primary amino acid sequence, which led me to ask the question, okay, like, could we learn what could we learn just by looking at these sequences on their own without any labels, in the self-supervised learning fashion that had been recently applied in natural language processing with transformer models. So in this self-supervised setting, for instance, if we take BERT, for a long time in NLP, we had models like maybe bidirectional LSTMs or ELMO that were developed for a particular task. So you learn the weights and the embedding at the same time as you were working on a task. That was, I guess, what Howard and Reuter did, and Reuter is presenting in 2 weeks now. But what was done in BERT is that they took multiple layers of transformer encoder units and stacked them together and learned contextual embeddings, not in autoregressive fashion, but in more contextual fashion, such that for any given token, depending on the on its context, you have a unique vector describing it in 768 or whatnot dimensions, and this was done in what is called the self-supervised fashion or semi-supervised, because you only put in primary sequences or just sentences, and some of them were masked out, I guess, because you didn't want to peek into the future tokens. And let's say if 10 or 20% of tokens were masked out, that one of the pre-training objectives was to predict the missing token, right? With this mass language modeling objective. And then you have some gradient coming from cross entropy loss, and also some gradient was also coming from what is called next sentence prediction task, which is basically asking whether two sequences that are in the model are consecutive, or they're not, or they come from a different document, which is quite a controversial nowadays pre-training objective, but I'll get into that a bit later. So this pre-training is done on millions of sequences, and then fine-tuning is done just for a few epochs, even on a small data set, and you can apply to a variety of tasks here, like in question answering or named entity recognition, which is an annotation task that you do along the sequence, or natural language inference, where you have some hypothesis and entailment, and you see if the hypothesis entails the premise in a two-sequence classification task. Yes, so knowing all of these transfer learning methods, I was curious to pursue the project, and I saw that I wasn't the first one, basically, to be interested in this methodology. And in this slide, I won't spend too much time here, but I just want to expose you that within the last 2 years, quite a few people were interested in modeling protein language, just because of its industrial applications, I guess, as well, and some big companies. And most of them used ELMO or bidirectional LSTMs. And most importantly, they used a very, very small vocabularies. So they tokenize at character level. As I said, there are around 20 or 30, depending how you look at it, amino acids, and they treated them as words, whereas they treated the whole protein as a sentence. So the training procedure seems quite simple. You pre-train on sentences that have words, and for me, they didn't look too straightforward, because in something like neural machine translation, you use at least 10,000 subword, 10,000 big subword vocabularies often, or much more. So I thought, okay, this could be an interesting thing to do. And some people also thought the same in parallel. Yes, so you can take a look at the slide maybe at home if you're interested in the field. So the first contribution that we did in my work is to get rid of this 20 amino acid vocabulary and introduce 10,000 subword vocabulary. And that was achieved by byte pair encoding algorithm that was resurrected by Rico Senrich, who's here at the University of Zurich. And the algorithm is quite simple. It's not one line, maybe 3 lines, but you initially start with several character level tokens. and then you sift through the training data set and you merge the most common, the most frequently occurring subwords together. And originally, I guess in the compression algorithm, they would give these two most frequently occurring subwords a new token to compress sequences, but here you just concatenate them together. And this algorithm is quite deterministic. So here we specify that we want 10,000 subwords to appear in our vocabulary, plus or minus. And as you can see for amino acids, like, yes, there is an amino acid called A, there is a C, cysteine, there is another one called D. So at the single character level, we get an encoding for these amino acids. We also get an encoding for the most frequent two-character amino acid combinations. So LL, AA, AL, VL, they appear very often in biology. So it could be useful to have them. And some of them are quite odd and weird, and these could be also sequencing errors as well. And now, if you take a look a bit deeper, like you'll see that this choice of using 10,000 subword vocabulary was quite rational, because we tested several different sizes and we observed that if you use 10,000 subwords, then most of the tokens are 3 to 4 amino acids long, which is useful because once you have 4 amino acids, then the first and the last can already interact in 3 dimensions, or at least we have this assumption. And as you can see here along this 10,000 word index, some of these tokens are extremely long and are very conserved in the nature. So this vocabulary building technique makes sure that we capture this diversity of the nature. Why it's useful as well, byte pair encoding makes sure that we compress our sequences by around more than 60%. So here in this figure, let's assume you know that we have a BERT-like model and we want to input 512 tokens. And unlike in machine translation or some other tasks, often proteins are quite long, as you can see here in green color. And it could be, around 18% of them are actually forming this right tail here. And they would not fit in the model if we tokenize them by character. However, after byte pair encoding compression, most of the sequences do fit into the model. So for something like single sequence classification task or 2 sequence classification task, it's quite a good idea to use BPE. Okay, so another contribution that we did, and it's not really a new theoretical contribution, but once again resurrecting, I would say, something that has been done before. Here, remember, in BERT, your pre-training objective is mask language modeling, and there was this next sentence prediction pre-training objective as well. However, you know, we decided to use Roberta model and just go with mass language modeling as a pre-training objective. But we still kind of thought, it would be interesting to find a biological alternative for next sequence prediction. So somehow find a relationship between 2 proteins. And I don't know, could anyone imagine such relationship for proteins? I don't know, probably in the chat, I can imagine there's 20 answers now. But one exemplary relationship would be, for instance, predicting if 2 proteins belong to the same family, to the same protein family. But that is actually achievable quite easily just by using multiple sequence alignment and using distance-based methods or hidden Markov models. I think the harder problem is to predict if protein A and protein B will bind with each other. So this is what we used as our next sentence prediction objective. So all of these models that I showed you previously in this table of protein language modeling that was done since the last 2 years, they would just take in protein sequences, like single sentences, like described here. However, we said, okay, let's take half of our sequences will actually bind. So the first one here is binding, and the fourth one is binding, and these two are not. However, we will not necessarily give them explicit labels if they're binding or not, but we'll just see if this BERT-like model can understand through mass language modeling alone why the two sequences come together. And for that, we modified Roberta architecture a tiny bit. So during pre-training, we put proteins that bind, like the first one and the fourth one and the third one. Here, this binding information came from string database that is maintained by von Mehring from University of Zurich as well, in his functional genomics research group, I think. And the other half of the sequences will just be random permutations of proteins. And then we'll separate them by the SEP token and put them in the model during pre-training. So I guess now we can see why this also byte pair encoding is useful, because we'll be training usually on two sequences, and we'll have to make sure that the sequences, the concatenated sequence after embedding, after encoding with byte pair algorithm fits into 512 token limit. For the fine tuning, we used three, I would say, subtypes of fine tuning tasks, and one of them, one of the types works at the sequence level, another one works at sequence pairs, so checks how the two proteins are interacting, and the third one is a token level classification task, or basically annotation task. Just like in named entity recognition, for every given token, we have to spit out some label. And I'll get into these tasks a bit later. So now we did the pre-training, and we could see that this model that I described, where we put in two sequences at once, we're just calling it strings, since from the string database, string to sequence model. On the holdout data set had the smallest cross-entropy perplexity loss, and compared to just single sequence pre-training. And we didn't do just one data set. We went quite wide in breadth, and we tested several other data sets that some of them had like more than 30 million sequences, and some were extremely small. And this training and optimization for hyperparameters took quite a while, but all of these pre-trained models are available, and I'll try to share the slides afterwards, and you can use them and build your own biological services or contribute. Yeah, and hyperparameters, maybe leave it for questions. And one sanity check for pre-training is to see if remember I told you that our vocabulary has 10,000 subwords, so we just pulled 20 that correspond to amino acids, and we did like t-SNE dimensionality reduction to see if we could recapitulate some of the biology behind these individual amino acids. So for instance, if you look at this in red, hydrophobic, aromatic are very greasy and big amino acids, and they occupy a lot of space, and here we can see that they clustered together. And the same happens with these greasy and the smaller amino acids, isoleucine, valine, alanine. And of course, you can also play with these parameters and cluster a lot of things, but we replicated this across all models and with UMAP as well. And now, I should say that, okay, so for pre-training, you can see a picture like this, or maybe you could also try to predict if the sequences are from the same family, but it's very hard to evaluate prediction performance, because as you can see here, you just get some perplexity score, but that is not too informative. The gold truth always lies in what can you do with this pre-trained language model. And for our fine-tuning tasks, here at they came in 3 different types. Like I said, just like from this figure looking at BERT, we had like single sequence classification, where the CLS token pulled all the information, hopefully, and could predict a class label. Then we had sequence pair classification task with 2 proteins, where also only 1 CLS token was participating in prediction, and for the sequence annotation task, just like in named entity recognition. So for these 3 types, we used 6 different tasks that are quite canonical. So for something like localization, which is a 10 class prediction, you have to predict into which subcellular compartment does the protein, where does it end up after expression? Does it stay in cytoplasm? Does it go into nucleus? Does it get embedded into the membrane, or it shoots out from the protein and roams around and does its business somewhere else? So for that task, we did extremely well yeah, and we beat the state-of-the-art result. And then for something like solubility, which is a binary class, whether the protein will fold correctly or not, will it precipitate or not, we didn't do too well. Something like homology prediction, which basically says, for a given protein, could you map it to 1 out of 1,200 different protein classes? We did quite well. Just I'm showing here accuracy results, but for more than 1,000 classification, it's quite good. And then another task was protein-protein binding, PBB. And that is exactly very similar tasks to how we pre-trained our model, because we put in 2 sequences that half of them were without any label binding and some were not binding. So for that task, we did extremely well. And all of these results come from this 2-sequence pre-training model. Whereas, for instance, for this protein-protein binding task, if we use only single sequence model, the accuracy was 0.92 or 93. So it was significantly less, but still quite good. Then for T-cell receptor, we had a data set prepared by Anna Weber from IBM, and where you're predicting for some particular epitope. So for instance, maybe a piece of coronavirus that enters your body and gets partially digested. It is just a tiny piece of protein that hopefully your T cells, your immune cells will learn how to recognize. And for a big fraction of us who are attending this talk, our immune cells are learning this actively maybe, or have learned it already. So here you're predicting, will this particular T cell receptor sequence bind to the epitope of the virus or bacteria or some other antigen? And at least at the time when we tested it, it did quite well. And I think now Anna has improved this baseline result by quite a bit. And for the annotation task, which is secondary structure prediction, where you put 3 labels along the sequence, we didn't do too well, and that is an ongoing task. And if I have time, I'll explain you what has to be done. Okay, so one thing, yeah, is to have this results, and once again, these are just numbers. Then we also aimed to be as transparent as possible to try to visualize the attention mechanisms. And if you use this BERT-like model with 12 layers, 12 encoder layers, and 12 self-attention heads, we thought it would be interesting to see where exactly for any given sequence or sequence pairs, where the attention is being paid and what the model finds significant when making predictions. So here is a dummy example. We have this rationally designed tin barrel protein that consists of 7 or 8 alpha helices and some beta sheets. And here we're taking just a single alpha helix as an example, and for more examples you can go to the repo. And after tokenizing this alpha helix, we get 8 tokens, beginning of sequence, end of sequence, and some actual useful information in between. And here we can see, for instance, that the first layer, or the first I would say 3 layers, mostly learn positional encodings or just positional relations between these tokens. So basically it's doing alignment. So in this example, the the current token is aligning to the previous one, and in some other example, the previous token is aligning to the current one. And then in the other layers, we say that, or traditionally, if you look at something like a BERT trained for a particular pre-trained on English, usually see that layers 4 to 8 usually encode some more dependency relationships, maybe syntax, so between the words. And here we can also see quite somewhat structured, yet still messy picture, but definitely something has been learned here. And then traditionally, the last 4 layers or 3 layers, when visualized, usually show some unique global patterns that are particular for that task, or pre-training or fine-tuning task. So for instance here, if we did in this figure, I can show you that, remember, we pre-train sometimes on 2 sequences, on sequence pairs, and sometimes on single sequences. And just comparing this pre-trained picture, we can see that pre-training on 2 sequences gives an interesting result, because the middle layer, the middle several layers are not really learning any of these dependency relationships, or they're really focusing on beginning of sequence token. However, after fine-tuning for protein-protein binding, we see that this model becomes much more structured, and starts paying attention to maybe more charged amino acids like K, C, and VQuil. So for instance, if you look, if you want, we can explore a bit more these layers. So in like layer 3, there are several patterns that you can see at once, like both alignment and paying attention from the beginning of sequence, just from the first meaningful token, not the beginning of sequence, but yeah, actual amino acids, yeah, to the last or to the previous one, which is charged. And similar patterns are observed in these layers, maybe, I guess, layer 6 if I have. So this layer 6 is more interesting because it has quite a few attention mechanisms, and that's where we say that dependency relationships are learned. So for instance, quite curiously here, for every token, as I go down, MD, KDE, AW, for each of those tokens, we're paying attention only to the end of sequence tokens. So here MD pays attention to these 3, then KDE pays attention to the end of sequence, and AW pays attention to end of sequence, and KC does as well. But then once you have this end of sequence, 3 last tokens, they start paying attention to the first ones. So that is an interesting dependency relationship where one half of the sequence pays attention to the other half, and we're speculating that this is exactly how you can predict protein-protein binding, for instance. And here, in a bit later layers, we see some global patterns. So for instance, where it forms kind of more close set where just the first few tokens pay attention only to themselves. They form this exclusive clique, the party for themselves, so to say, and whereas the very few last tokens also interact only with themselves. And also there is often, yeah, we saw this extremely strong signal always to beginning of sequence or end of sequence. Yeah, so I guess it was quite a short talk. I could talk a bit more about some future directions that I find exciting, but for now I can show that what we found here is that we were the first people to try to use this byte pair encoding algorithm to create a subword vocabulary somewhat rationally that has some biological meaning. And in doing so, we reduced the protein sequence space by 64%. That for some reason hasn't been done before. We also pre-trained Roberta on 2 in several different ways, with single sequences or 2 sequences with, also we used Roberta to initialize longformer pre-training because they're quite related. So we heated up longformer from our learned Roberta embeddings, and now longformer model has that sliding attention window that if you can set the size to the same 512 tokens, and you basically have several Robertas in one model, and we pre-trained it for around 2,000 token sequences, and here you could use such model for multiple protein interaction prediction, for instance, or for some extremely long protein like Titan, or I don't know, whatever weird protein happens in your body. And we achieved somewhat good results for these other tasks, and our paper was luckily accepted also to machine learning for the structural biology workshop at NeurIPS. And we're sharing all of our code, like pre-training code and scripts, how to pre-process the data, and the pre-trained models, and the raw data is also shared at IBM Box. So if you're curious, definitely go there, make a pull request, and yeah, if you want to do something else with these models. And maybe quickly, I could just show you where the field is heading, at least, you know, from how I feel about it. So one thing for sure, and I see it often in the literature, that people are working with quite long documents, not only sentences, right? Most of machine translation, for instance, is also done at document level, right? Or document summarization. So if we want to predict binding between multiple proteins, we'll have to have models that somehow deal with this problem of having quadratic attention, computational cost in BERT-like models, by making some sparse approximations to it. And recently there were quite a few studies that use like locally sensitive hashing, or some other methods. So those could be quite interesting to explore. And I guess as attention is getting cheaper and cheaper, we could put more and more sequences into the model. Also, we encountered this problem that I'm also trying to work a bit on my free time after my thesis, is to make sure that we can use for annotation tasks, that we can use this 10,000 subword vocabulary, because, so for instance, in this structure prediction task, we have 3 labels, 0, 1, and 2. And along this sequence, depending how we tokenize the sequences, sometimes these tokens, by using byte pair encoding, will break the boundaries of what we call these label islands that are continuous. So here we have 5 zeros, but our tokenization algorithm here cuts them correctly. However, at some point it might not. So here I'm just imagining an ideal scenario, but how to enforce it is much harder, because you don't want to touch the testing data set. And another thing is that quite a few people at IBM are also dealing with attention models, maybe more for protein to small molecule binding, or small molecule binding, and or chemical reactions. And here they developed this algorithm, RxN mapper, that can give you some probabilities for binding. So I was also thinking of adapting this model where for a given protein binding site, you could get probabilities of other protein binding there. Yeah, and so to wrap up the talk, I would like to acknowledge my supervisor at IBM, the ever so excellent Matteo Manica, who's just a master of many things, including programming, statistics, machine learning, and whatnot, and team management and leading. I learned a lot under him, and Yoris for all sorts of help, and for interest in protein modeling as well. The group of computational systems biology at IBM is led by Dr. Maria Rodriguez, and if you're interested in any super, I don't know, tough, I would say, biological questions that can be solved by computers, definitely contact her. And at ETH, Professor Karsten Borgward helped me out as well.